Right. This is the second part of the transcription regulation audio-video handout, and in the first part we focused on the DNA in this figure from our textbook, and in the second part we're going to focus on the proteins. Just to review the DNA part, this is a story for any gene. The DNA part, the DNA of any gene is composed of two parts: the transcription unit that codes for RNA. So the transcription unit is DNA. And the DNA gets transcribed into RNA, the primary transcript. And this is the structural or business, the functional part of the gene. So, for example, in our example, the insulin gene, this is the information that includes the code for the insulin protein, and that's called the coding region. So the coding region is within the part of the DNA that gets transcribed. It's included in the coding region is included in the exons of this gene. Okay, and we'll review exon and introns in another context. So one part of every gene is the transcription unit. The transcription unit is made of DNA. The other part of every gene is called the promoter, and the promoter is composed of two parts: the basal or proximal promoter, and the basal or proximal promoter tells RNA polymerase, this green ball, where to land and start transcription. So the proximal promoter is always right next to the start of transcription. This is the start of the transcription unit. This is the start of the place that's where the DNA is going to get copied into RNA. So this is a basal proximal promoter. It's the proximal promoter because it's right next door. It's the basal promoter. Another term that some scientists call this is the minimal promoter. It's the basic or minimal promoter. It just has enough information to show RNA polymerase where to land. But it doesn't tell the RNA polymerase how often to land there and where to land there, in what cell types. Here's one thing that the author has highlighted here in this figure, and that is this DNA sequence has a special name. It's called the Tata box. And when we move on, we'll see that the DNA of the Tata box has transcription factors, these green balls, that are bound to it. So proteins assemble on the basal promoter, and they get RNA polymerase there and help position it to start transcription. The second part, and I should just mention that this part of the promoter is boring because it is basic. And in developmental biology, we really want to know, what gets insulin turned on in the pancreas and stay keeping it off in all the other cell types. And that's the job of these enhancers up here, which, as we talked about in the first video, are maybe more appropriately called response elements. And we'll see in this part of the video that the response elements are the same in every cell type. What's different is these activators that bind to the response elements. So let's just get a clean slate and talk now about these proteins. The relevant proteins are RNA polymerase. This is the enzyme that transcribes the gene. So this is the actual job of all these proteins. This does the job of all these proteins. These proteins are all regulating whether or not RNA polymerase shows up on the gene and moves down the gene. One of the concepts of transcription is that RNA and proteins that get made from it are not particularly stable and get degraded. So if we say that the insulin gene is on in the pancreas, we mean that it's kind of always on. That for a gene to be on, it has to be copied again and again. So if we looked inside the pancreas cells and we looked at the DNA for the insulin gene, in those cells, at a pretty high frequency, RNA polymerase is landing at the start site and transcribing this gene. And once one gets going, another one lands. And it's just a assembly line of RNA polymerases landing and transcribing the gene. So that's the business of gene expression or activation of genes, is to get RNA polymerase to land there. In all the other cell types in our body, there's no RNA polymerase here because the gene is off. So the gene is on, we mean that RNA polymerase is actively transcribing the gene and repetitively transcribing the gene. All these other proteins are involved in determining whether or not RNA polymerase is being delivered to the gene and how often. So the where, when, and how much transcription that occurs on this gene is the job of these proteins. And these proteins are all called transcription factors. And they can fall into two categories. These orange sort of goldfish guys are called transcription factors. And some of them are activators and some are repressors. And as you can see here, activators and repressors bind to the DNA, the purple DNA of the enhancer or response elements. So these are transcription factors here, and then these blue guys are also transcription factors. The first guys are called activators. They're all transcription factors. Repressors. These guys, the blue guys, the best name for them is co-regulators, because for these guys we have some co-activators and some co-repressors. Okay, so we've got co-regulators, and that term's defined on the handout, and we've got activators and repressors. And the difference between these is that activators and repressors bind directly to the DNA. And then these co-regulators don't bind directly to the DNA. So all these proteins are transcription factors. We've got activators and repressors that bind to the DNA, and then these co-regulators that don't bind directly. How do the co-regulators get to the gene? They bind to the tails of the activators and repressors. When we go and we look at these activators and repressors, let's just clear this off. These guys all have, these are all proteins, and these guys all have two parts to each protein. That part is called the DNA binding domain. And the DNA binding domain binds to a specific DNA sequence in the enhancer. These are typically about 14 letters long, 14 nucleotides long, plus or minus a couple. I mean, they could be from 12 to, say, 20 nucleotides long. So there's a specific sequence of letters here, like something like this. You know, just a sequence of letters, and the DNA binding domain of this promoter binds directly to that DNA sequence. So the DNA binding domain is often called a DNA sequence specific binding domain, because it's going to bind just to those sequences. And because of that, what we'll see is that we'll see transcription factors that are bound directly to the promoter of the insulin gene in the pancreas. The biggest difference between the gene of the insulin gene in the pancreas and in other cell types is, in the pancreas, these proteins are all present, and in other cell types, these proteins are absent. The reason these proteins are getting assembled here is because of the DNA binding domain of these activators. These activators are only made in the pancreas, or maybe only active in the pancreas, and in being made and being active, they bind to this sequence and they assemble all the other stuff. All the other stuff assembles just because it sticks to the tails of the transcription factors, and so through non-covalent interactions, these proteins stick together. That's one part of these transcription factors. So all activators and repressors have a DNA sequence-specific binding domain, and they also have a transcription activation domain. Another kind of poor term because some are activators and some are repressors, but it's typically called the transcription activation domain, or it's called the transacting domain. Okay, and so that's this part here. That is the transcription activation domain, and that's another part of the protein, and that's the tail of the protein that's sticky. In some cases, it's sticky, in this example, to some co-regulators. In other cases, it might be directly sticky to RNA polymerase, and because that's the whole purpose of the transcription factor, is to regulate the binding of RNA polymerase to the basal promoter and the onset of transcription. So we've got this set of transcription factors that binds directly to the DNA, and then we have this other set that can be co-regulators. Some of these co-regulators, those proteins, some include enzymes. Some co-regulators are enzymes that modify other proteins. For example, RNA polymerase has a long carboxy terminal tail that gets hyperphosphorylated, and these co-regulators regulate the phosphorylation of the RNA polymerase and regulate its movement into the transcription domain and the transcription of the transcription unit, like that. It's not shown here, but some of these enzymes in these co-regulators actually modify other proteins. And they can either kind of clear the way downstream for RNA polymerase to move down, get the job done, and get the gene transcribed. They can sometimes sort of open up the gene by modifying other proteins, or sometimes they can kind of close down the gene and make it inaccessible. And that's a really important part, and that's one of the big differences between the gene, the insulin gene in the pancreatic cells, is that the gene is opened up and ready for transcription, and in other cell types it's closed down and transcription is blocked. And so to kind of think about this, again, the promoter is something that developmental biologists are really interested in, because the promoter regulates where, when, and how much of a gene is expressed. And that's really a key factor in causing cells to become different during development. So we start out with a single fertilized egg, but over time, as cells divide, we start getting cell types that are pancreas cells and brain cells. And how they become different is they express different genes. So the promoter is really important in factoring that. But again, remember that the DNA from the promoter is the same in all cell types. And the difference is really whether or not these transcription factors are present in the cell and are active in the cell. And these are the dynamic part of the gene and the dynamic part of turning genes on and off. And we'll stop there.